Chapter 16, Part 5. B lymphocytes. These guys do what is called humoral immunity. They make antibodies. So this part of the immune system, the B lymphocytes or B cells, and the antibodies they produce are found in the spleen, in lymph nodes, and in mucosa-associated lymphoid tissues, so in the mucosas. So some of the B cells circulate in the blood. Most of them do not circulate. They are tucked away in the spleen or the lymph nodes or the lymphoid tissues, and their major function is production of antibodies. So these cells have a B cell receptor. That should sound familiar. The B cell receptor is a bit like the T cell receptor. It's a protein. It's on the plasma membrane of the B cell. It has a constant region and a variable region, and it's composed of 4 proteins held together by disulfide bonds, very similar to the T cell receptor. There's a couple of differences. Oh, and it also has antigen binding sites. This one has 2 antigen binding sites. The T cell receptor has 1. It has part of it that touches the MHC protein, and then it also has the epitope binding site. It has 1 piece. The B cell receptors recognize the same antigen, but they recognize 2 of them potentially simultaneously. So a particular B cell makes 1 kind of B cell receptor. So each B cell receptor has 1 epitope that it recognizes, but it has 2 sites to bind to the same epitope. All right. So you have a whole collection of B cells with each of them having specific B cell receptors. And that is your collection of antibodies that your body can produce if those B cells get stimulated by antigen, by finding their epitope. So let's look. This is the structure of the B cell receptor. It's anchored to the plasma membrane through a domain of the protein, a piece of the protein, extension of the protein that penetrates through the plasma membrane. Out here we find the 2 pieces. So this part here is part of the variable region. This is variable region. These 2 are the same on every B cell receptor. They're both identical. They recognize the same structure, the same epitope. And there's 2 of them, so it could recognize, if there was another one of these, it could recognize it here. So there's this transmembrane portion. And then we have this part that's on the outside of the cell. And again, it's held together by disulfide bonds, these different peptide pieces. But this cell, all of its B cell receptors have the exact same antigen binding sites. But different B cells will have different antigen binding sites. So they'll recognize different epitopes. When a B cell finds its epitope, it does mitosis, and it goes through mitosis several times, many times, and it produces a whole bunch of what are called plasma cells. The plasma cells circulate, but they are all genetically identical to that B cell. They are going to make the protein, the B cell receptor epitope, the antigen binding site that recognizes the epitope is going to be the same on every one of those plasma cells. And what those plasma cells are going to do is they're going to make B cell receptors, but they cut off the part of the protein that holds it to the plasma membrane of the cell. So these guys make these B cell receptor proteins, but they aren't stuck to the surface of the cell. They float, and we call those antibodies. Let me say that again. Let me go back 1 slide. There we are. So here's our antibody. What happens in the plasma cells is they cut the proteins right here. And it's the same, all the rest of this is the same. And so they release a huge amount of this kind of protein, and it circulates in the blood, it circulates in the plasma, it moves through into body tissues, into interstitial fluid, and into lymph, and they will stick to whenever they find their epitope, they hold on to it, and they stick to it, and they don't let go. So this cell, this is a plasma cell. So this cell's whole job is to make proteins that get secreted. The only function it has is to pump out thousands and thousands and millions of antibodies. So they are a massive secretory pathway protein synthesis. So this is all of these membranes in here, these are all rough ER. So we are doing translation with thousands of ribosomes and just huge surface area of rough ER, and then Golgi body. And so all of those proteins from all of this ER is moving to the Golgi and getting secreted and released out into the interstitial fluid to become part of the proteins in the blood. So these guys are where it's at in terms of antibody synthesis. These are the ones that actually make the antibody. And each one is genetically identical to the cell, the B cell it came from, and it recognizes the same epitope as that B cell did. So B lymphocytes and antibodies have specificity. I've just been talking about this, and antibody structure. So these are immunoglobulins. The antibodies are called immunoglobulins because they're in the blood, they're in the plasma, and they are similar, as I said, to the BCRs. They are secreted by activated B cells called plasma cells. They have the identical specificity as the B cell receptor. And here's the structure of them. And actually, this is a protein map of how the structures of the proteins. So they really do look like a Y. They have a little hinge point here in the constant region, and the variable tips out on the end, which is where the epitopes bind. So the function of the antibodies. So we know that they can stick to epitopes. What are the effects of what happens when they stick to epitopes? So they do a bunch of different things, and that's what makes them so useful. We've talked about complement, and we talked about inflammation. Antibodies are one of the ways to get complement activated, right? That's the classical. And they're also involved in initiating inflammatory events. Neutralization. We can neutralize things like toxins, protein toxins. If we make an antibody that sticks to the toxin, in the part of the toxin that has to move a little to do something, or that has to touch something else in order to function, and now it's got an antibody sitting on it, it blocks it from being able to do that function. It can't touch whatever it has to touch because this antibody is physically in the way. Antibodies can prevent viruses from being able to infect by sitting on the protein that the virus needs to come into contact with a host cell protein. So that's neutralization. Opsonization is another thing, and we already mentioned this also because complement does opsonization, and whether it's complement or antibodies, and often it's both, this is covering a thing, protein, a bacterium, a virus, whatever it is, covering it with antibodies or complement proteins, just coating it with proteins that are involved, that phagocytes, for example, have receptors for the constant region of antibodies. That's the part of the antibody that sticks out if there's an epitope, on a virus or on a bacterium. The epitope binding site, the antigen binding site, is going to be pointed toward that thing, and what sticks out of the antibody is the constant region. Then the neutrophil can grab that part of the antibody, so anything the antibody has. So it helps with phagocytosis. It helps with a bunch of different cells' ability to get hold of and trap and engulf those cells. They also kill by oxidizing. They cause agglutination. This is when they clump up. I got images of some of these in a second. And we also see antibody-dependent cellular cytotoxicity. So let's look at the diagram here. So this is neutralization. These proteins literally just physically block the cells from being able to do stuff. Opsonization coats things, and there's a receptor here for constant region of the antibodies, and that allows it to grab hold of it, and now the bacterium can't escape. Agglutination is clustering because each antibody can bind to two antigens. They are able to grab two at the same time, and we get this agglutination process going. Sometimes these can cause oxidation just by binding in the presence of oxides. We can get oxidation of the bacteria, and we can get lymphocytes involved here sometimes. A lymphocyte in the area, natural killer lymphocyte, will recognize that the antibody is stuck because of that receptor, the constant region is sticking out, and it will cause the cell to die. It will release a protein called perforin that perforates, punches holes in the plasma membrane of this pathogen. There are 5 types of antibodies produced. They are made in different parts of the body. They are released in different ways, and they have some different functionality. So this gives us some diversity in ways that we can defend our bodies from pathogens. So they vary based on what the portal of entry they're defending is, and the kind of function that is necessary. So we've got 5 classes of antibodies. The first kind of antibody produced when we first encounter a new epitope, a new antigen that we hadn't seen before, is IgM. Those are the first ones made. The next ones made, and then the ones that are made most of the time after that, are IgGs. These are also the longest lasting. So these guys are most common in the blood. If you look at plasma, it's mostly IgGs. IgA is found in body secretions. Basically, if it's wet and it comes out of your body, it probably has IgA in it. So tears, saliva, vaginal secretions, nose secretions, lots of milk. Milk has IgA. IgE is one that's made in response to parasitic worms and also to allergies. And so we find that people who have allergies often have elevated amounts of IgE antibodies. IgD, this is the least common, and it's also the one about which we know the least. So I like this image, mainly this figure here. This is important. So this is the IgM. IgM, this is the first one we make. It is a pentamer, right? There are 5 antibodies, and they're held together at the center with a bunch of proteins that anchor them to each other. This means that this can bind to 10 different antigens with epitopes at the same time. So these are fantastic for agglutination. They're big, they're bulky, and so anyway, we make these. So this is at the beginning of an infection. We're making lots of this. This is IgG. The structure is just a base. This is your basic antibody, the Y shape with the epitopes at the epitope binding sites, antigen binding sites at the tips. This one is IgA. IgA can sometimes be a monomer. So it's often a dimer, so that there's 4 antigen binding sites, and they're all made in the same cell, so they're all the same antigen that they recognize, the same epitope. This is IgE. This has some differences, but it's basically a similar structure to the IgG. This is IgD. The other big differences between them is how long they last, how many days they hang around. Some of these don't last very long. You make them and then they're gone pretty quickly from the blood. Others last quite a bit longer. So, similar to the T cells, we talked about clonal deletion in the T cells. The same kind of thing happens to the B cells. This happens for them, it happens in the bone marrow, but it's a very similar kind of process. The B cells that are self-reactive either get inactivated or they change their B cell receptor. Sometimes they can do that. But then they have to still go through this screening process again. But they do that rather than go through apoptosis. So a lot of them do go through apoptosis, but a few of them do have these other options. This is, again, just like with the T cells, this is to prevent your body from producing antibodies against your own normal cell components. So again, we're going to make randomly just a bunch of B cells, each with its own unique B cell receptor. Some of them will bind to autoantigens, and so they will go through apoptosis. Some of them, as we said, we have those other options, but most of them will actually go through apoptosis. A few of them will not react with autoantigens, and so they will be able to go to the spleen ultimately, finish developing, and then wait for their antigen to show up. And when it does, that's when this cell will become a plasma cell, or do mitosis and produce a whole bunch of plasma cells, and then produce lots and lots of antibodies. In the next section, we're going to talk about cytokines.